Okay, so where do we start? We start with sucrose. Will sucrose go out? Why not? It's got no sucrose outside. That's very good. Read the instructions. Sucrose can't cross the membrane. That's what it means. Okay. What about sodium? Which way does it go? Follows its concentration gradient, doesn't it? That's what passive rules say. High to low concentration. The mantra in your brain. Passive. High concentration to low concentration. Always. It's always dependent on that. And glucose? Yeah, I didn't say anything about glucose, so it can go through. Okay? So just remember I can make up whatever rules I want here. So read the instructions. Good, so we write what? Sodium go, I guess they both go in. Sodium and glucose in, is that right? Yeah. Just at the initial point, right? Just at the beginning. And then what about water? What do we do? Okay, well we have to show our work, right? So what do we have to figure out? If water follows its own concentration gradient, according to passive rules, what do we need to figure out? Where do we have more free water? Good, so we add up our total osmotic concentration or total solute concentration. And it's higher, wait, it's higher outside, so that means we should go in, doesn't it? No. Good. So remember, this is solutes, so it's higher outside solutes. And again, the volume's the same. Somebody's making fun of my picture up there. Okay? I'm just kidding. It's higher solutes outside, so that means more hydration shells around each solute. More hydration shells means less free water. Remember that? So more solute, less free water. So that means we have more free water on the inside here, comparatively. And so that means it's going to come out, and the cell's going to shrink, isn't it? At least initially. Everybody agree? So osmosis, we get net osmosis out of the cell. Okay, did we already define the hypertonic versus hypotonic? Yes. Which solution is hypertonic? The cell or the environment? The environment. Okay, good. So this is hypertonic. So you either say the environment is hypertonic to the cell, or the cell is hypotonic to the environment. Right? We're always comparing two solutions. Good. Want another one? Okay. So I'll just make up some more numbers. Okay. Let's see here. We'll make it up. Make it up as we go, right? We'll leave sucrose the same. And we'll say, let's see, we'll say 2 there. How about that? So 2, 2, 3, and 1. Go ahead and do the same thing. So I love this question. All I have to do is change the numbers, and you get a whole new situation. Woo! So for those garbage cans. Having fun yet? I'm going to add the salt. Okay, so let's see. Sucrose doesn't move. Sodium? Sodium goes out this time. So you say sodium goes out. Sodium out and glucose in, isn't it? Yes. So now they're going in opposite directions. And then I did this on purpose. What happens with osmosis? Okay, here's the trick question. Does water stop moving? No. Okay, good. So you see no net osmosis here would be one answer. Or water, you see no change in water movement because water is moving back and forth evenly or at equal rates. So you don't see any change in the volume of the cell, at least at the beginning, okay? So these are then isotonic, is the term. These solutions are the same concentrations, basically, and so you don't get any net osmosis across the membrane. And again, what was the point of this? I think I told you already, didn't I, with the cells, plasma, right? You don't, the 0.9% sodium chloride you get in an IV closely matches the solution in your plasma. So it's closely isotonic to your plasma, your blood plasma, so that when you get an IV, it doesn't hurt your cells. They don't swell or shrink. Okay, so can you guys do one of these? There's a 110% chance you're going to get one on the exam. Okay? 110. Yep. That means it's almost certain, right? Okay, any questions on that? Pretty straightforward, maybe? And again, over time, the concentrations will change, so then the water will start moving. But for all these questions, we always start at the beginning time point, okay? Just to make it simple. Questions? Answers. Does anybody have any answers out there? Why are we here? Why is the sky blue? It's refraction. Yeah, so good job. Okay. Okay, let's finish off this chapter with just a little bit about active transport, because you need to be able to compare and contrast passive and active, okay? Can I erase this stuff now? Okay. So passive didn't rely on a direct input of energy. That's going to be the major difference between passive and active. I think this is letter C, if I remember correctly. Passive was letter B. In your notes. Active transport needs a direct input of energy, and it's almost always going to be ATP, which we'll learn in the next chapter is the energy currency of the cell. Okay? So it almost always needs a direct input. Passive was dependent on a concentration gradient, which is a form of energy, but it's not ATP. Okay? Because of that, it's almost always, not almost, it is going to always be facilitated because you're going to have proteins that use this energy to do work, okay? So there's different types. There's uniport, where you move things one way or the other across the membrane. You can have symport, that means two things moving in the same direction at the same time. or antiport, they're moving opposite. But all these show an enzyme doing this, okay? Because the enzyme is going to use the energy, use the ATP to do work. But why is it necessary for this energy? Most of the time, oh, did you want to answer? That's right, that's right. It's going to go against the concentration gradient for whatever solute we're moving. So we're going to see that these enzymes move solutes from low to high concentration, which goes against the rules, doesn't it? What does the rules of physics say in passive transport? Always high to low. We're going to learn anytime we break the rules, or I can't really break the rules, but go against the rules in the cell or in our body, we're going to have to do work to do that, okay? And work here means you need energy. There's a whole chapter on this, okay? But we're going against the rules. Passive says high to low. Active, we're pretty much always going low to high. And a lot of the times what you'll see, we do that to actually maintain a concentration gradient, actually build a concentration gradient. The classic example is the sodium-potassium pump. Sodium-potassium pump. It's an antiporter. I didn't hear you, sorry. It's an antiporter that moves sodium in one direction and potassium in the other, each cycle. And it uses ATP to do that. And if you look closely at this figure in the book, you'll see that typically sodium levels are lower in the cytoplasm and higher in the extracellular fluid, or the outside of the cell. And potassium is the opposite. And yet, this sodium-potassium pump will move the sodium and potassium both against their concentration gradients. This, of course, is for a reason. We'll talk about that in a second. But it does this using energy from the ATP. Okay? We'll learn that ATP has these high-energy phosphate groups, which you can use to do work, which enzymes use to do work. Yes, sir?
So how does the enzyme decide that sodium is going to go out and potassium is going to go in? It looks like the side of the potassium is smaller, or the sodium is going in, and is it going out the potassium part larger?
Yeah, yeah, you're right. There's a size difference in the sodium and potassium ions, and that means there's a size difference in the hydration shelf. And so the enzymes will see that difference. So again, we're going to learn that enzymes are specific for what they do. So this enzyme will not transport anything other than sodiums and potassium. And they have unique binding sites. It is a really amazing enzyme. So here's kind of what it does. And this is kind of cool. This is an example of an enzyme changing shape or conformation. A lot of them do that while they do their work, whatever it is. But in this case, it's going to bind 3 sodiums. They're actually smaller, so you can bind 3, and uses ATP to then change shape and move the sodiums across the membrane. Remember, this is a transmembrane protein, so it's going to have access to both sides. But it kind of looks like a Pac-Man, doesn't it? Or Pac-Woman? I don't know. I can't tell the gender of this, okay? So there is no gender with proteins, as it turns out, okay? So it binds 3 sodiums, spits them out this side, and notice we go from low to high. And then once it's open this way, so also the shape has to do with this as well. The shape change of the enzyme affects its affinity, if you will. And this conformation, then, it has a higher affinity for potassium ions, and you can only fit 2 because it's bigger. But anyways, it binds 2 and then changes shape again to transport those. And the potassium is going from low to high. So it goes from low to high, okay? So you're not going to have to draw this, but this is a cool example because when you get into physiology, and we'll come back to that. Oh, I don't have the picture in this. When you get into physiology, you're going to learn that neurons are very, very dependent on concentration gradients. Please let me see. I have the picture here. There it is. Neurons create what's called an action potential down the axon. You'll learn all about this in phys. But that's dependent on a concentration gradient of sodiums and potassiums. You'll learn that as that signal goes, you know, it starts in your brain, goes down your brain stem to your spine, and then out to your wherever. That's how your muscles move, right? That's all conducted through the membrane of the neurons. And what it is you'll see this rush of sodium ions across the membrane, which then produces this potential down the whole neuron. But then what has to happen is those sodium-potassium pumps have to kick in really, really quickly and pump the sodiums back out so that you recreate that membrane potential. Okay? So it's really, really important for neurons to do that. Remember I told you that most cells don't let things go to equilibrium? That's pretty much death. Same thing here. Our neurons can never go to an equilibrium. Otherwise, that's bad news. You know how nerves control everything, your movement, breathing. Here's kind of a morbid example. You guys into like forensic files or anything? Not really? Okay. Have you heard of rigor mortis? Yeah, rigor mortis is after somebody dies, their body can get really contorted for the first, I don't know, 20 hours or so. After that, sorry. Well, yeah, then after that. But the reason why they get all contorted is because the muscles aren't allowed to relax. That's because this action potential is required to be active for the muscles to relax, so to actually not be contracted. But if you run out of ATP, will the sodium-potassium pump be working? No, no. So after you die, hopefully not for a really long time, okay, right? We're all young here. But after death, you run out of ATP, so then the neurons aren't working, so then the muscles are all contracted, and they stay that way until the body starts to degrade more after death, okay? So that's kind of a morbid example, but remember, I think I mentioned at one point, cells spend an absurd amount of ATP every day just doing maintenance activities, like 10 to the 30th ATP. And a lot of that is just maintaining concentration gradients. So really boring stuff, but we're really happy that it does it because it's kind of required for life, yeah? And it's a good thing we don't have to think about it either. Because just think how quickly my neurons have to reset the concentration gradients every time I move my muscles. It happens very, very quickly. It's kind of cool. This picture here is just showing more examples of how proteins can change shape. So that channel changes shape, which then affects how it actually transports things, okay? And what it can transport. So it's kind of cool. We'll look at some more examples later on of proteins that change shape. Okay, so what do you need to do? You have to be able to compare and contrast passive and active to each other. Yeah?
So when the proteins change their shape, do the polypeptides also change shape?
Well, proteins are made of polypeptides,
Right.
so.
They don't change.
Well, they both change. So it's the amino acids actually moving around.
Okay.
They're not detaching from each other, but the protein is a 3D shape, so parts of it can change.
Okay.
Yeah, it's really kind of cool. If you get bored, you can get on YouTube and put in protein conformation changes or something. I don't know. It's pretty nerdy, but it's kind of cool. Okay, so passive. What is passive dependent on? Hammer it into your brain. Always have to have a concentration gradient. It's going to go high to low, no matter what direction. If it's high on this side and low on this side, then it's going to be, if it's simple, what kind of molecules would it have to be? Small nonpolar. Very good. Very good. Small nonpolar can do simple diffusion. If it's charged or polar, then it's going to be facilitated. And even some of these carriers can change shape without ATP. So it's kind of cool. These are all dependent on concentration gradients. And then active, well, I have my list up here, right? First of all, you look for that use of ATP. It's almost a dead giveaway that it's active. 99% of the time, it's going to be facilitated. It might be 100% of the time. And that's because why? You have a protein using energy to do what? Moving from low to high. So we're going against the rules, and you have to have energy. Can't rely on a concentration gradient to do that. Okay, that's pretty much it. That's pretty much it. So there's not much more with active transport. So I won't ask you any really detailed questions about the study of the plasma. Any questions on that? Expectation wise? Okay. Sorry, I always give you examples from cells, but it, makes it more interesting, doesn't it? When you see real life examples. I think it does. Okay, anything else? That's pretty much it for chapter 7. Chapter 7 was lipids, compare and contrast lipids and membranes, and passive and active transport. Okay, are we ready to go over the study guide now? Okay, let's do it. So again, you'll find the study guide in the modules section. There's 2 files there you should be looking at. The study guide, obviously, and then the study guide for cell components. I can move this over a little bit. So, starting way back in chapter 2. So, with chemistry, you have to know how to describe an atom, right? That's how we started out. Got a little bit of that on the quiz. So what do protons and electrons do for you? Okay. They give you charge plus protons tell you what element it is, right? Okay, so I'll work with that. Let's see, neutrons, the only thing we asked about that was atomic mass and change. You have different types of different numbers of neutrons, right? So if that shows up. But you don't have to do any calculations. So you won't have to calculate the atomic mass or anything like that, so it's nice. Let's see, bonds. What are the 2 types of bonds that we focused on? Covalent and ionic. You need to be able to compare and contrast that. Many of you lost points because you failed to mention that covalent is stronger than ionic. So if the question asks, compare or contrast, you want to look at the differences as well. So just be aware of that. Use the keywords, like sharing versus ionic as a transfer. Right? What is an ion? Something that has a charge, right? An imbalance between protons and electrons. Let's see what else. Molecules, yeah. So molecules are made up of elements, right? What are polar covalent bonds? So comparing polar and nonpolar. And what's the result of polar covalent bond? Hydrogen bond. Hydrogen bond is not about filling your electron cell. It's the result of polar covalent bond. And it was key to all of the properties of water that we talked about. You guys have your polar and nonpolar bonds memorized? Should. What are the polar bonds that we see? Seen that. Hydroxyl, yeah? What else? CO and NH. Yeah, you should have them memorized by now. Those are all polar. And then non-polar? CH and? Yeah, anything bonded to itself, right? I always put OO, it doesn't matter. Put nitrogen next to each other, it doesn't matter, right? Anything bonded to itself. So remember, it's the difference in electronegativity. You should know what that is and why that leads to polar bonds and what, or if it doesn't, okay? I think you should have that memorized. If you guys have been studying, you should have it memorized. I told you to do it several weeks ago, yeah? Hopefully you're on it. Okay, anything on chemistry? I forgot. Yeah, I know. Okay, water. So we talked about the properties of water. There won't be any questions on adhesion or cohesion because we didn't talk about that in class. It's in the book, but not in class. Acids, bases, buffers. Okay, you don't need to do anything scary like you did on cell concepts with the buffers. Okay, that was an application of buffer knowledge. You just need to know the general definitions of what they do. What is a buffer? Resist pH change. How? Acts as an acid or a base. Well, you have to know what those are. So what's an acid? Anything that increases hydrogen ion concentration. And a base is anything that decreases it. So a buffer can do both. Cool. You can either absorb hydrogen or release hydrogen, depending on what's being added to it, right? So just think about how they work in general. You don't have to do anything nasty like I gave you. Okay? That's basically it. And you should know the pH scale versus hydrogen ion concentration. That still can be confusing for students. So the number goes up, that actually means the hydrogen ion concentration is going down. Right? With an inverse relationship, you can still mess that up. Get it down, you don't miss these stupid points. Okay? I think that's pretty much it. I think I already mentioned the water properties comes from that hydrogen bonding ability, right? That comes from polar covalent. So just remember, there can be other compounds that are covalent, polar covalent that can interact with water. Right? Like sugars. Okay. Chapter 4. So I gave you a whole list on why carbon is cool, right? Go back and review it. It has what, 4 openings in a code? Sorry, 4 openings in this valence shell. You can form lots of bonds, or up to 4 bonds. And we can form bonds with lots of different things, right? We don't see that with silicon. And we do see carbon chain, right? So I think I gave you, that was one question on the quiz that tripped you up, I think. The common characteristics of macromolecules. You always see a carbon chain, and then functional groups. All these functional groups. You guys got your functional groups memorized? Yes. I'm picking on you now. Am I still on the screen? So let's see what else. I already gave you one. What's that? What's this one? Okay, there needs to be more than a couple of you. Come on. How about this one? This one, nobody gets the spelling right. Carbonyl. Carbonyl. Try to spell them right on the exam, too. Okay? I'm going to give you a molecule with a couple, at least 3 or 4 functional groups. Try to get them right. That's carbonyl with an N. Carboxyl has that OH, carboxylic acid. So get a memory. It's pretty good. You guys did good at the perfect. Okay. Questions on that? Yeah. Sulfhydryl is an SH. Yeah. If you see SH, it's sulfhydryl. Oh man, I make fun of you for spelling, but I don't think I can spell that one. It's like sulfhydryl, something like that. That's an SH. Right, covalent bonds. Yeah, that was on the list, wasn't it? On one of the lists, at least. Yeah, we'll see it sometime. Yep. Oh yeah. Oh, you guys are gonna get so sick of me. Even after we're done with this exam, we're still gonna do that. So we'll write down this, and you should say. It's over here. CH bonds. This is non. This is polar. So that's polar. That's polar. Right? You guys are gonna get sick of me. I already know it. You're not sick of me yet, right? Don't answer that question. Okay, what else from, where are we? Chapter 4, functional groups. And so don't get caught up in all the enantiomers and all that other stuff in chapter 4, but just remember isomers. So what's the difference between cis and trans isomers? Yeah, so I would just use the fatty acid examples we looked at, right? So a cis unsaturated fat is heated or bent. Trans are more linear, right? Because of where the hydrogens are. Just use that. Just remember isomer is change shape or conformation, even if they're the same chemical formula. Yeah. No, no, the cis and trans are only associated with what kind of fats? Unsaturated. So if I said it wrong, I'm sorry. Cis and trans are both unsaturated, means they have to have a double bond in them, right? But the trans sometimes act. Well, the trans act metabolically like saturated. So saturated or linear, they don't have a double bond. The trans are more linear, even though they have a double bond. That's what I was trying to convince you of. Way to yeah, so that's a common thing students mix up, the saturated and unsaturated. When we're talking about cis and trans, we want to make sure it's talking about unsaturated fat. Okay, anything else on 4? That kind of went into 5 as well. So for 5, we just looked at sugar and lipid structure, right? So can you compare and contrast a triglyceride and a phospholipid? Just in general, not going to have to remember a specific one. Huh? No, a triglyceride is not a sugar. No, triglyceride is a three-tailed lipid, 3 fatty acids. So it's triglyceride and phospholipid are the two. So a triglyceride, remember tricycle, versus phospholipid. So here, this phospholipid only has 2 tails, and it has that phosphohead. So that's in chapter 5 and 7. And then with sugars, remember sugars are that ring structure with a bunch of OH groups on it, right? That's all you pretty much have to remember. So highly polar. Well, you should recognize a disaccharide is 2 sugars stuck together, 2 monosaccharides stuck together. And then a trisaccharide and then polysaccharide is the long chain. So yeah, you should be getting used to the terminology. So mono means those monomers that you build things out of, right? And polymers are going to be the long chains of whatever. Yeah, you should know glycosidic bonds are the bonds between sugars. That's all you really need to know. So each sugar will have a glycosidic bond between it on the polymer, right? And then, yeah, dehydration and hydrolysis. All you need to know right now is that one forms polymers and one breaks polymers down. Right? So if you can't remember, try to remember hydrolysis. Lysis means to cut. So hydrolysis is the cutting one. You cut them down. So then that means dehydration has to be the one that's building them up, right? Building them up. So find a way to memorize it. We'll see it more as we look at other chains, like acids and proteins. We'll come across those terms again. But for now, you just need to know the general mechanism. Okay, anything else? Chapter 4, chapter 5? Got time, so we're doing good. Chapter 6 was a lot of memorization. Right? Can you compare, contrast prokaryotic and eukaryotic? Compare and contrast animal cell and plant cell. These those Venn diagrams. We already talked about organelles. So you write a sentence on each organelle. Which organelle sorts and packages protein? Good. Golgi. Ribosome makes them first, then they're sent usually to the Golgi. Not always. Think of the Golgi as the post office. They don't really mess with the packages, but they send them where they need to go, right? Yeah, so you got to memorize all those. Go back to the group activity we did, right? There's a few on there that I might have to pull off. So, let's see what else. Cell structure. There won't really be any questions about cell structure on that. just to start to understand. Right? That's all. So yeah, 6 you kind of breezed over, but a lot of it's just memorization. And hopefully it'll come back quickly. We had it within the past 5 years. Okay, anything on chapter 6? Again, that list of organelles and cell structures within a module is a cell component study. Then 7. So we already talked a little bit about phospholipid versus triglyceride. Fluidity, there will be a question on what fluidity is, what can affect fluidity. Talked about that a lot. I might ask you about winter wheat or something silly like that. Yeah. We need to know what things affect them, what it means, why does the cell care about having the perfect fluidity, whatever that is. Don't worry about types of membrane proteins. There won't be any questions on that. Just remember, all transporters are transmembrane. Always look at that. So I won't give you a big list of that. And then with passive and active, all you have to do is be able to contrast those. I won't even ask you any questions about sodium potassium bonds. But osmosis, that's confusing. That's that whole free water with hydration cell water. You need to really think about that. Draw your own little cell collection. Take yourself through the steps, okay? That can be confusing, figuring out where the higher free water is. Just based on what I know. Okay, anything else? Yeah. Oh yeah, we didn't talk about it, but there was one slide. It's where the cell can actually use a concentration gradient to do work. So if you look at the slides again, and this one right here is the example. You can actually build a concentration gradient and then use it to transport something else. That's coupled. They're coupled together. But I won't ask you about this. This is a cool example, though. This example is from the gut, small intestine, where you're digesting and bringing sugars across the membrane. They use a concentration gradient. So we are going to start new material on Wednesday. For the next chapter, just to stay on target. That won't be on the test, all right? Okay, good? Okay, good luck. Keep up the good work.